Let's take a minute and think about the anatomy of a single muscle fiber. Remember, we said that muscle fibers are surrounded by a specialized plasma membrane. The reason that this plasma membrane is unique is because it is capable of transmitting electrical current. As we'll talk about in a few minutes, the skeletal muscle is in close communication with the nervous system, and it is nerve impulses, or what we call action potentials, that are going to signal the skeletal muscle that it's time to contract. So in order for that signal, that electrical current, to travel from the nerve to the muscle, the membrane around the muscle has to be able to conduct electricity, and that's a special kind of membrane. In fact, neurons and muscle fibers are the only two kinds of cells that are capable of that, or that are capable of transmitting electrical current along their membrane. So we give this specialized plasma membrane a name. We call it the sarcolemma. Now, in addition to being functionally quite unique, the sarcolemma is also structurally unique in that along the length of the muscle fiber, the sarcolemma folds inward in regions called transverse tubules. Now, most people refer to these as T-tubules, so that's what I'm going to call them. T-tubules occur at regular intervals along the length of the muscle fiber. Again, these T-tubules are just areas where the sarcolemma folds inward, creating a little bit of a tunnel or a pathway that we call a T-tubule. Now remember, this membrane is capable of transmitting electrical current, so each T-tubule is there to help direct nerve impulses, not just along the length of the muscle fiber, but perpendicular to that also at regular intervals. It allows for electrical signals to be spread very rapidly along the entire length of the muscle fiber all at the same time. I'll show you a picture of this in just a minute. Now, in addition to these T-tubules, if you were to go underneath the sarcolemma, just underneath the plasma membrane, into the cytoplasm, you would find that specialized reticulum, the sarcoplasmic reticulum. So the sarcoplasmic reticulum is organized so that on each side of each T-tubule, there is a tunnel, if you will, of sarcoplasmic reticulum. And we call those terminal cisterna. That word cistern or cisterna can represent a cavity or a tunnel. Now remember, we said the sarcoplasmic reticulum stores and releases calcium ions. That's its function in the muscle fiber. And the calcium ions are going to help regulate the contraction of that muscle fiber. So if we were to travel down the length of a muscle fiber, every so often at regular intervals, we would find the plasma membrane, the sarcolemma, folded inward on itself in a T-tubule. And if we were to go across the sarcolemma into the cell itself, on both sides of each T-tubule, we would find a passageway of sarcoplasmic reticulum called terminal cisterna. So each T-tubule has two terminal cisterna associated with it. And together we call these three things, one T-tubule, two terminal cisterna, a triad. Now let's take a look at this unique anatomy. On this slide, you're looking at a muscle fiber. The muscle fiber has had its plasma membrane, its sarcolemma, cut away, so we can look inside. So this yellow structure right here is just the plasma membrane that surrounds this cell. But again, it's a specialized membrane, so we give it a name. We call it sarcolemma. This is an odd-looking cell, right? It's just an elongated linear structure. And when we look inside into the cytoplasm, most of what's in here are these big brown structures. These are myofibrils. Here's one myofibril. Here's another. Here's another. Here's another. Now, there are other things in here. You can see some mitochondria squished over to the side. There are going to be several nuclei in here. All the organelles are in here. Golgi apparatus and lysosomes and all the rest. But there's also a specialized organelle shown in blue called sarcoplasmic reticulum. Now, they're showing us that the sarcoplasmic reticulum surrounds all of the myofibrils together as a group, but it also sort of dives in between each myofibril. So there's sarcoplasmic reticulum all throughout and surrounding all these myofibrils. And that's important because the sarcoplasmic reticulum is involved in regulating muscle contraction. It's going to release calcium when it's time for each one of these to start contracting. Now, I want to draw your attention right here. You can see that the sarcolemma right here has folded inward, creating a little passageway that we call a transverse tubule or a T-tubule. It gets the name transverse because it's not running lengthwise along the length of the muscle fiber. It's running perpendicular. So it's transverse. It's a transverse tubule or a T-tubule right here. So that's what you're seeing in yellow, running over the surface of all of these myofibrils together. Now, the other thing you can see is that there are T-tubules all down the length of this cell, and they are occurring at regular intervals down the length of the cell. Now, the other thing you can see in the picture is that the sarcoplasmic reticulum, again in blue, sort of lines up with these T-tubules on each side. So this region right here in blue is called a terminal cisterna. This one is a terminal cisterna. And everywhere that you see one T-tubule with a terminal cisterna on each side, that's called a triad. So there's a triad right here, there's a triad right here, and right here. Now, when we see anatomical features, anatomical structures in a cell, there's always a reason. There's a reason why these triads are here. Remember, the purpose of the T-tubule is to help deliver an electrical signal, not just down the length of the cell, but across the cell, perpendicular to the cell. So the electrical signal travels down these T-tubules all down the length of the cell. And what that signal is going to help do is release calcium from these terminal cisterna. So when the signal arrives to this cell, we're going to have electrical signal traveling here and here and here and here at essentially the same time. So we're going to get calcium release all down here, all down here, all down here, all down here at essentially the same time. The entire length of this muscle fiber is going to be stimulated to contract all at once. So in order for us to understand how muscle functions, we really need to go down to that fundamental unit of contraction, the sarcomere, and we need to examine the anatomy of the sarcomere. We said that each sarcomere is made out of thick and thin filaments. Now, all a thick filament is a combination of a protein called myosin. So many myosins come together to make what we call a thick filament. The thin filament in muscle is made out of many copies of a protein called actin. Those actin molecules all come together and make a thin filament. Now, there are other what we call associated proteins, especially around these thin filaments. But the two primary players in a sarcomere are the thick filament made out of myosins and the thin filament made out of actins. The thin filaments are arranged around the thick filaments in what you can think of as a hexagonal pattern. In other words, it's important not to think about sarcomeres as being two-dimensional structures, even though that's how we draw them. It's actually a three-dimensional structure. The thin filaments surround the thick filaments in 360 degrees. On this slide, up at the top, we're looking at a single myofibril. Remember, there would be lots of these myofibrils inside a single muscle fiber. As you travel down the length of the myofibril, you see this very regular pattern showing us sarcomeres, one after another after another. The striping pattern is created by the arrangement of the filaments inside this myofibril. There are thick filaments and there are thin filaments, and they kind of overlap each other in a very special arrangement, creating a sarcomere. These lines that you see here in between each of the sarcomeres, those are the borders of the sarcomeres. They're called Z lines. So from Z line to Z line, that's one sarcomere. Now, if we were to cut through this myofibril, if we were to cut through the transverse plane, if we were to make a cross section, in other words, and then look at that cut face, it would look like this. This is an actual image taken with an electron microscope of the cut surface through a myofibril. So you're looking at a cross section through sarcomeres. Now, everywhere you see the open circles, those are the thick filaments in cross section again. And all the other sort of structures in between, the more solid looking, slightly lighter gray structures, that's a cross section through a thin filament. So you can see that around each one of these open circles, there are like spokes in a wheel. Those are all thin filaments arranged around each thick filament. You can pick any thick filament and you'll find 6 thin filaments around it. So you get this sort of honeycomb look, this hexagonal arrangement of thick and thin filaments. Now, when the sarcomere was first described, when scientists were first examining muscle tissue under the microscope and describing the sarcomere, they used some language based on what they saw under the microscope before they knew what each structure was actually made out of, what proteins were actually inside the sarcomere. So they came up with language including a pattern of banding and then a pattern of lines and zones within the bands. Again, this language was based on what they saw. They saw a dark band in the sarcomere that they called an A band. Then they saw a light band next to that they called an I band. And then within the bands, again, they saw this Z line. They saw what they called an H zone and an M line. Now, we now understand what these bands and these lines are created by. For example, we know that this dark A band in a sarcomere is actually the place where the thick filaments are overlapping the thin filaments. There are many, many filaments, in other words, overlapping each other, and that creates that dark appearance. The I bands, the lighter colored bands, are actually the places where there are no thick filaments, only thin filaments overlapping each other. The Z lines are also present in the I bands. Now, the other structures, remember the Z line. The Z line is found within the I band. This is the place, we now know, where the thin filaments are attached to something. The thin filaments attach themselves to this Z-line structure. It's simply a combination of proteins that create the Z-line. The H zone is a light area in the center of the A band, that dark band, and the M line is just a line in the center of the H zone. I'm going to show you a picture now, an actual image of a sarcomere, and we'll look at all of these regions. Now, it's important that you know not only where these regions are, but what we now realize they're made out of. So make sure that you can sketch and label a sarcomere. On this slide, we're looking at a drawing of a sarcomere, and we're looking at an actual sarcomere underneath an electron microscope. So let's start with the drawing at the bottom. This right here is a Z line, and over here, this is another Z line. So from here to here, from Z line to Z line, that's one sarcomere. Now inside the sarcomere, there are thick filaments, those are shown in brown. There are thin filaments, those are shown in blue. And then there are some other accessory proteins. For example, these Z lines are made out of protein. And there are other proteins in here that we can't really see. This M line is made out of protein. So again, there's other proteins involved, but the primary proteins are the ones that make up the thick filament, that's myosin, and the ones that make up the thin filaments, that's actin. So if you go up to the top, to the actual image, you can see what the scientists saw all those years ago. They saw this line here, and then one here, and they called that the Z line. And they said from here to here is a sarcomere. And in that sarcomere, they could see dark, and then they could see light. There's a light region here around the Z line. There's a light region over here around the Z line. And then there's this dark area here with a light area in the center. So from here to here, that's the A band. From here to here, that's the I band. And of course, there's another I band over here. Now let's think about what those are made out of. The A band, the dark band, is the place in the sarcomere where, if you go down, you have thick and thin filaments overlapping each other. Now, in the center of the A band, there's a light area that they called an H zone. And then there's a dark line right in the middle. The H zone is right here. The H zone is where you only have thick filaments. There are no thin filaments here. And again, you've got this structure made out of protein in the center called the M line. If you look at the I bands, They're an area where there are thin filaments but no thick filaments. And of course, the Z line is down the center. The Z line, made out of protein, is where these blue thin filaments anchor themselves. They're all attached. All the thin filaments are attached to the Z lines. Let's think about these thick and thin filaments now. We said that thick filaments are actually made out of multiple copies of this protein called myosin. So there are many individual myosin proteins that come together and bundle up into a thick filament. Now, when you look at a thick filament, it's got a really interesting arrangement of the myosins, and that's because of the way myosins fold. Remember, all proteins fold up into a unique three-dimensional shape, and that shape gives them their function. So inside a thick filament, about half of this bundle of myosin proteins are arranged in one direction, and the other half is arranged in the other direction. So you have two mirror halves to a thick filament, and they meet in the middle. Myosin has a region within it that is referred to as the heads. And this arrangement, this mirror arrangement of all these myosins, creates a thick filament that has heads sort of jutting off of it at 90-degree angles. And those heads are going to protrude all the way around the thick filament in 360 degrees. And again, at both ends, on both mirrored halves. Now, I know it's hard to visualize this, but again, I'll show you an image in just a moment. This pattern of the heads of the myosin molecules jutting off of the thick filament around 360 degrees is critical because the heads of the myosin are going to need to come in contact with the thin filaments. Remember, and the thin filaments are arranged around the thick filaments. When the heads of the myosins bind to the thin filaments, arranged in that hexagon pattern, we're going to create a temporary bridging that we call cross bridging between the thick filaments and the thin filaments. On this slide, up at the top, you're looking at one thick filament. So remember what this is. This is a whole group, a whole bundle of myosins, all joined together. But they're joined together essentially in two parts, in mirror images of each other. So this is a bundle of myosins with the heads all on this end. And this is a bundle of myosins with the heads all on this end. And there's this linear region in the middle that has no heads coming off of it. So there are many, many, many myosins that make up this thick filament. Now, if we were to take this little region right here and blow it up, this is what it would look like. So each myosin is essentially this linear region with two heads sticking off at the end. And as you can see, there are many of them in here. And they're arranged so that these heads are sticking out all around the 360 degrees of the thick filament. Now, it's not shown here, but remember, arranged around this thick filament are thin filaments. And they're also arranged in 360 degrees, all the way around. So this arrangement of the heads is important because we're going to need these heads to come in contact with the 6 thin filaments that are surrounding this thick filament. Remember, structure and function are tightly connected to each other.